Ladies and gentlemen, good morning. Welcome to the third Avatar Medicine Forum. I'm your host for today, and I'm delighted to have this opportunity to meet with our audience once again this year. This year marks as the third year of our Avatar Medicine Forum, and our goal, as always, is to deepen everyone's understanding of the new concept in Avatar medicine for patient treatments. In 2023, it has been a remarkable year for AI technology, significantly transforming various industries and taking Avatar medicine to a new height, seamlessly integrating into various aspects of human life. Let's welcome our first speaker, Luke Dimasi, the Senior Product Manager of Emulate, a biotech company specializing in organ-on-a-chip technology. Luke has over 15 years' experience in the field of life science and biotechnology and will share his expertise with us.
Hello, everyone. My name is Luke Dimasi. I am the Senior Director of Product Management here at Emulate, and I'm really excited today to be here to speak to you about Emulate, what organ chips are, and how we're using this technology to drive next generation in vitro modeling for preclinical discovery and drug development. I'd like to thank the organizers of the Avatar Medicine Forum for inviting me today, also to speak to you and introduce you to some of the exciting science that we're seeing that's generated on our platform. Before I get into Emulate and what organ chips are, I thought I'd take a step back and just reflect on why Emulate is here. So we believe, and it's not just us, but also our customers have told us that the drug discovery and development process is fundamentally quite inefficient. And really this is costing not only pharmaceutical companies in terms of the lost dollars that are spent bringing a drug to market, but because it is inefficient, it's also impacting society as a whole. So, why is this the case? And really, when we take a look at the numbers, 46% of drugs that enter clinical trials fail due to efficacy reasons, they just don't work. 26% fail due to toxicity and safety concerns. And despite decades of life science tools advancements, the cost to bring these drugs to market is in excess of $2 billion and is only increasing. So why is this the case? And again, our customers have told us that it is the preclinical models that are used before these drugs get into clinic. And really, it comes down to two things. Conventional models simply just cannot recreate the complexity that of the human body and also the human specificity as well. So when we take a look at these models, they fall into two buckets. There's the conventional in vitro models. These are usually cell lines, can be 2D, sometimes 3D cell culture. But typically these types of models lack biological complexity. So you're not able to model the full range of responses that are seen in the human body. Animal models are another way of testing, and they are complex, and you're able to see different types of pathways and processes, but they are not human relevant. And we have seen that this has caused a problem in translation when we take these drugs and test them in animals. There are other factors here that are also influencing the status quo. And first, because of the increasingly targeted and human-specific nature of drugs that are coming out these days, there's been an increasing reliance on non-human primates as they have biology that's more similar to humans. However, we know that there are shortages with non-human primates, and we also know that there is increasing scrutiny from regulators as well as society as a whole on the use of these types of animals for testing. Another factor that's influencing preclinical models is the regulatory environment. And right now, there's been a couple of changes that have happened in the regulatory space. The FDA Modernization Act that was passed last year here in the US, as well as the EU Animal Testing Action Plan that are now opening the window for pharma companies and academic scientists to start to try to investigate and use different types of methods for testing both efficacy and safety in drug development. If we zoom in on the in vitro cell culture space, we can see over the last few decades, there has been a number of step changes on the path to creating more human relevant biology. When in vitro cell culture first started, decades and decades ago, typically cancer cell lines or even primary cells were the main types of cells that were cultured in vitro. And these cells are good for testing very specific things, pathways, specific pathways, specific yes/no questions, but they typically lack complexity. A couple of decades ago, the industry started to embrace 3D cell culture with the introduction of steroids, mainly tumor steroids. Here, now we're starting to play in a realm where microphysiological environment of what happens in the body is actually being replicated in vitro. And here we're talking about things like oxygen gradients or even nutrient gradients, and also contextualizing these cells in a 3D environment. In the last decade or so, we've also had a couple of other step changes. One, the introduction of iPSC technology, which now enables us to model a far more greater range of cells, and also takes the burden off of only using primary cells, which are hard to come by, also have lot-to-lot variations, so very difficult to draw conclusions on. And then finally, in the last few years, there's the organoid movement, whereby we're taking a multitude of cell types and putting them together, enabling them to differentiate, and you're able to see that complexity in a contextualized three-dimensional environment. So as you can see, we've had a number of step changes over the last couple of decades. And we believe here at Emulate that organ chips are the next step change. You can essentially take any of these biological inputs, and then once you put them on a microphysiological system like an organ chip with the relevant microphysiological forces like flow and stretch, you start to see how the biology changes. It begins to differentiate. It becomes to function a lot more like it would function in the body. I'll take you through some examples a little bit later. So Emulate, we believe the past is using static and non-human preclinical biology, but the future is introducing dynamic and complex human biology to the bench. And we believe organ chips are a way of doing this. In our system, we take a human-centric approach to building our models. We use human-relevant cells, either primary cells, iPSCs, or organoids, and any of these starting biological materials can be adapted to culture on chip. The chip itself models a cell-to-cell and a tissue-to-tissue interaction, so we enable multicellular complexity. Typically, most of our models have an epithelial or parenchymal chamber, as well as a vascular interface. We introduce tissue-specific microenvironments such as flow and cyclic stretch. And again, this recreates tissue-relevant mechanical forces that cells feel in the body. And this pushes them to differentiate and function a lot better than static or even two-dimensional culture. And organ chips are fundamentally quite flexible. You can model a wide variety of organ models. You can add in complexity like immune cells, microbes, hypoxia, and Emulate has a number of workflows that enable our customers to do just that. So for those of you who aren't familiar with Emulate and organ chips, I'm going to take you through the two chip designs that we have on market today. The first one here is the chip S1. This was our first chip that we launched a few years ago, and it has both stretch and flow as part of its feature set. If we take a look at a cross-section of the chip, you can see that there are two chambers. We have a top chamber, 1 millimeter by 1 millimeter, and a bottom chamber, 1 millimeter by 0.2 millimeters. Typically, cells in the top chamber are epithelia, and cells in the bottom chamber are vascular. These two chambers are separated by this flexible and porous membrane, as you can see. And you're able to culture cells in each channel independently, so you can see them independently. We have vacuum suction on chip that enables you to stretch that biology. This mimics peristalsis or breathing. And you're able to independently flow fluids through each of these chambers suited to the cell types. And you're able to take effluent off of that biology so you can see how it's changing over time. So you get a lot of data off of an organ chip. So that was our chip S1. This year, we actually launched our second chip design, the chip A1, and this is our accessible chip design. And this chip really enables you to model different types of biology on the human emulation system. So the chip A1, just like the chip S1, has two chambers. We have this top chamber here that is accessible when you open the lid, and you're able to cast gels and seed cells in gels. This top chamber is interfaced with a serpentine endothelial chamber. And it is our belief and hope that our customers now have the flexibility to begin to build more complex three-dimensional models on this A1 chip that you weren't able to do on the S1. Because the chamber is open, you're able to have that direct access to that biology, so you can expose that to drugs through aerosolization or even topical delivery. So again, two different chip designs that we have available and suited for a variety of applications. So let's talk for a moment about the actual organ models that Emulate has developed. So we have currently 4 organ models on market. 2 are for the intestine, the colon intestine chip and the duodenum intestine chip. We have a liver chip and a proximal tubule kidney chip. In development, we have 2. We have the airway lung chip as well as the brain chip as well. And these are suited for a wide variety of applications. So for colon intestine chip, we're able to model the complexities of things like inflammation, IBD, and immune cell recruitment. For our liver chip, we're able to accurately model and predict drug-induced liver injury. With these models that Emulate has built, we have characterized the models, we've provided application validation, and we guarantee reproducibility because we provide not only the organ chips themselves, but also the pre-qualified cells and biology that goes on that chip. So really, it's an organ chip model in a box. But that's only the beginning. Outside of the organ models that Emulate has developed, our academic community and organ chip community have over 100 publications that describe over 70 types of organ models, everything ranging from lung, alveolus models, to bacterial co-culture, bone marrow, kidney, glomerulus, lymphoid follicle, as well as colorectal cancer models on chip. So we're seeing an ever-expanding menu of organ models that our community is building on our platform. So why organ chips? And here we believe that with greater complexity comes greater predictivity. And by predictivity, I mean that we are able to predict what happens in the human body when it's exposed to a drug or a treatment. And we went about actually proving this with one of our organ models, the human Emulate liver chip. So the liver chip itself is a human-relevant model of the liver sinusoid. It's a primary human model. It has multicellular complexity. This includes primary hepatocytes in the top chamber. And then in the bottom chamber, we have stellates and Cooper cells, as well as sinusoidal endothelial cells as well. And this makes up that liver sinusoid organ unit. With all these cells coming together with relevant flow, we see enhanced hepatic functionality. We see the ability to produce albumin. We see the response to drugs improve, gene expression improved. And we've validated this chip for, again, toxicology, or specifically drug-induced liver injury. And again, we not only want to show that the model is more complex, but that this complexity drives a high level of predictivity. And one of our landmark publications that we published last year actually set out to do just that. We took 27 known hepatotoxic compounds as well as their non-toxic structural analogs. And these drugs weren't just picked by Emulate. These were actually chosen by an independent consortium of pharmaceutical companies that really wanted to pressure test microphysiological systems and really validate them. So we took that list and we threw it through, we tested these through 3 donors on a quite expansive human liver chip screening campaign. What we found was that organ chips were able to correctly identify 87% of the drugs that cause drug-induced liver injury. So all these drugs actually got to market, and the ones that were on market and toxic, so essentially animal studies, picked them out as non-toxic, the liver chip was able to identify 87% of those as toxic. Which is great in terms of its sensitivity. But not only that, we wanted to benchmark that against the best in-class 3D hepatic steroid tests or screens that are on market today. And we showed that that these types of 3D hepatic steroids only come in at 47% sensitivity. So we're seeing almost a doubling in predictive power by adding this complexity. What we also described in this publication is that if you were to have this increased predictive power, what does that mean in terms of improving the efficiency of drug discovery? And the publication goes from a model whereby it was calculated that the industry itself would save about $3 billion in productivity gains just for implementing organ chips. For an average pharmaceutical company with an average size pipeline, this translates to about $150 million a year. So again, here, this publication really does set out to prove that you can quantify the impact of complexity through predictivity as well as the financial reasons and benefits to a pharmaceutical company. Liver is one of our models, but as I mentioned, we have a rich and diverse organ chip community out there. One of our academic partners at the Ellison Institute, and this is Dr. Mamanthola, has published on development of a colorectal cancer on-chip model, and really studying the microenvironmental influences that occur in the body on the metastatic spread. So in traditional cell culture is such as transwells or other 2D cell cultures, or even 3D cell cultures, you're lacking some of those microphysiological forces that normally occur in the body, such as flow and stretch. Here we showed that these are important for really understanding the mechanisms and uncovering new mechanisms of how cancer metastases occurs and how it progresses. So in this publication, the Ellison Institute developed and characterized a colorectal cancer on-chip model. Here they developed a KCO2 model in the epithelia, and they seeded cancer CRC organoids on that model, and they were able to see how those invaded and then also invaded and underwent intravasation into the endothelial compartment. They were able to demonstrate that there's that the metabolic comparison between intestine chips alone versus intestine chips with the CRC colorectal cancer cells on them. And they were able to show and demonstrate that the metabolic profile matched what actually occurs in vivo. And also they were able to examine the tumor cell intravasation on chip, and they were able to show that the chip was able to more accurately reproduce or recapitulate what actually happens in vivo versus simpler static models. Conclusions of the study is that CRC exhibits phenotypic heterogeneity during intravasation, and the mechanical and biochemical cues from the tumor microenvironment do impact this invasion profile and heterogeneity. So here they basically showed that stretch, peristalsis, is impacting and a mechanism for selecting how these cells actually metastasize the gut. I would encourage you to take a look at the Emulate website, and there's a wonderful webinar that was done by Dr. Mamanthola that explains this in detail. Another example, and this was published in 2021, is again, when you have organ models that are more physiologically relevant, they express the right types of genes, you're able then to test them for looking at advanced or complex mechanisms of toxicity. And one of those is looking at on-target off-tumor safety assessment of certain cancer drugs, such as TCBs, T-cell bispecific antibodies, or other MABs. So in this publication, we established a human immunocompetent organ-on-chip system. I think, and here they looked at two models in particular, the colon model as well as the lung. And they were looking at a specific target that was expressed on tumors, but was also expressed at low levels in normal healthy tissue. the organ chips themselves did recapitulate that expression of the target at vivo relevant levels. And the system was able then to be used as a screening tool to screen different TCB constructs to see which ones were able to still hit the tumor but not activate immune cells in normal or healthy tissue. And again, you can only do this when you have a model that is able to recapitulate that complexity and that transcriptomic profile. Just shifting gears here from cancer and toxicity, another area where organ chips have utility is in inflammatory bowel disease. This is a complex disease. There's multiple reasons and multiple pathways that are involved here. 81% of drugs that try to deal with things like Crohn's disease fail in clinic due to efficacy. The ones that do get past clinic and get into market, half of the patients fail to respond to the therapy, and of those that do respond, 90% of them fail to enter long-term remission. So inflammatory bowel disease, again, one of these complex diseases that we still cannot develop drugs to be able to have long-term remission and effectivity. So we're able to model this quite nicely on organ chips with our human intestine chip, the colon chip specifically. And let me show you how we built the colon chip and how it models this inflammatory process here. So in the colon chip, we use primary derived colonic epithelial cells, and this is complex with colonic and microvascular endothelial cells. When we start to model the inflammatory process, we prime the chip with a priming stimulus. This starts the inflammatory process with the endothelial cells. They start to express these adhesion molecules. We flow immune cells through that vasculature, and we see them rolling along that endothelium as they would in the body. They eventually bind and then move into the epithelial compartment. And then once there, they carry out their effector function. They express cytokines. These cytokines then have an effect on the epithelia, and we see the epithelia start to degenerate, and a leaky gut or a leaky pathology or barrier disruption starts to occur. So this is a complex sequence of events that we're able to actually recapitulate on chip quite nicely. Some examples of the data we're able to generate here, and you can see the movement of the immune cell from the endothelial compartment into that epithelial compartment. We're able to quantify what that looks like through confocal microscopy. When you label these cells with the cell track, you can see that we can quantify cells that stick to the endothelium, the underneath. We can see cells that are starting to migrate through that membrane, and we can see cells that actually get on top into the epithelial compartment, and we can quantify each of these. And we can also see that without priming here, that we get very little migration of the cells, but with priming, we see quite a substantial improvement there. The biggest thing here, which is important, why organ chips matter, is that to recapitulate this biological process, which normally occurs in the body, accurately in vitro, you really need that flow and that necessary shear through that endothelial channel. That provides the necessary selectivity, the gate, that only the right types of immune cells bind and then actually make their way into the epithelium. Without that flow, what we see is that everything binds, and you get a very non-specific migration of cells, and that's not what happens in the body. We've also been able to validate the model with a number of different anti-inflammatory drugs with different modalities. We've tested corticosteroids like dexamethasone, which is first-line treatment for IBD, but we've also tested targeted therapeutics such as AGN-300, which is an alpha-4 beta-1, alpha-4 beta-7 molecule that blocks that interaction, and also TNF-alpha blocking agents as well, like Cimzia and Humira. We see that all of these are able to knock down that immune migration, the recruitment, migration, and then that effector function. But essentially what we're doing here is we are modeling a very sophisticated way of looking at the immune cells. So again, testing a broad immune cell functionality in a human relevant tissue. We are able to see selective immune attachment. We're able to see that immune migration into the tissue. We can quantify effector function and cytokine release. As I mentioned previously, you're able to look at the effluent over time, so you can see real-time changes in that biology. And we can see also real-time changes in the tissue damage or dysfunction. And this is what we've shown with the immune cell recruitment for IBD or inflammatory modeling. But this is also a good way of testing other types of immune cell functionalities, including engineered cells like CAR T. Now, CAR Ts really are a godsend in terms of treatment of certain types of cancers. Been very successful in the treatment of blood cancers. However, the issue with these types of cells is that they don't work well in solid tumors, and unfortunately, 90% of adult cancers are solid tumors. And the reason why is a multitude of reasons. First, that these cells, these cells, once they are infused into the body, they lack the ability to traffic to the tumor in an efficient way. And then once they're there in the tumor, we see the tumor microenvironment applying immunosuppressive factors, which really dampens their ability to kill tumor. So although they show great promise, there is still a lot of work that needs to be done to optimize these types of modalities for solid tumors. So what we wanted to do is we wanted to try to apply this immune cell recruitment workflow to look at how we could better characterize the ability of these CAR T cells to traffic and migrate into the tumor and kill target. For that, we developed an experiment, a workflow. We selected CAR T cells. These were second generation HER2 and CD19 CAR T cells. And we also looked at untransduced controls. We administered these CAR Ts through the tissue-specific vasculature. As I mentioned, we have that vasculature compartment, and we had relevant shear, which drives that selectivity. And we sourced these off the shelf. Emulate is not a CAR T developer, but we were able to partner with ProMab, who had these ready-to-use CAR Ts available for after use. In the top chamber, our target cells were an A549 HER2 positive non-small cell lung carcinoma cell line. And again, this was seated in the top channel. This is really just to show that we are able to capture the killing and effector function of those CARs once they traffic in. And then we wanted to see, once we have this established, how can we then test different co-therapeutics to see if we can modulate or improve that response. And in our proof of concept testing, we've looked at IL-2 preconditioning as well as STING agonist, which are two relevant mechanisms of improving CAR T effectiveness in vivo. So here first, let's take a look at the migration, the recruitment of the cells. So again, a lot of data here, I'll step you through it. But first off, when we look at just applying that first column there, HER2 cells alone, you can see that there's without priming, there's very little recruitment of the cells into the epithelia. When we prime the chip with relevant pro-inflammatory cues that would normally occur in the tumor, we can see that we can increase that recruitment of those HER2 positive CAR Ts. We found that by treatment with IL-2, pre-treatment with IL-2, pre-culture before we added to chip, the CAR T cells that were pre-treated were able to recruit a lot more vigorously than those that weren't pre-treated. The STING agonist itself really didn't do anything in terms of the change to recruitment or migration. And we saw the same type of pattern with CD19. Again, like at this stage, we believe that both HER2 and CD19 would have the same response in terms of the recruitment and migration. And we definitely saw that here. So this was an interesting readout, but what really started looking even more interesting is when we looked at the ability of these cells to kill. So you can see here what we're looking at is caspase signaling in the top chamber. And you can see that the control as well as the HER2 without priming, so without getting the cells in there, very little caspase activity and very little killing occurring in that top chamber. HER2 cells with priming, you can see we're able to get cells recruited into the epithelium chamber, and we're able to see a subsequent increase in caspase signaling. we definitely saw an increase in caspase and signaling and killing with IL-2. But you would recall from the previous slide here that IL-2, really the mechanism here that we're seeing is that increases the ability of the cells to actually migrate, so motility of the cells. So this is probably a factor of just more cells being in the top chamber. The interesting mechanism here is that STING, although STING didn't change the recruitment of CAR-Ts, it certainly changed the ability of these CAR-Ts to kill. You can see that here by the jump in caspase signaling with STING agonist as part of a co-therapy. And just to show the specificity of response, because the target cell line was HER2 positive, the CD19 negative control really didn't show much killing at all. In fact, very close to control. So here what we're showing on chip is that we are able to successfully see recruitment of CAR T cells, and we're able to profile different mechanisms of functionality of that cell with these two different types of co-therapies. IL-2 seems to increase the ability of these cells to recruit and migrate. STING agonist seems to increase the ability of these cells to kill. So the profiling different types of mechanisms. So when you're looking at developing different CAR-Ts, you can look at different strategies to improve the effectiveness of these. And on chip, you can start to see that with the complexity. Just wrapping up, one final example here, and this is done on our new Emulate A1 chip, our accessible chip. And this is proof of concept data. It was actually published this year, in fact. Researchers at the Wyss Institute were looking to model Barrett's esophagus and look at esophageal adenocarcinoma progression on chip. So how does the interaction of CAFs and fibroblasts have on the epithelial, and they wanted a good system to test that. So what they did, first of all, this was a first-in-class in vitro model to assess the heterogeneous response of Barrett's esophagus epithelium to co-cultures of different stroma and fibroblast cells that were resected from tissues of varying disease states. So they looked at co-culture of these cells with normal fibroblasts, metaplasia fibroblasts, as well as dysplasia fibroblasts, or CAFs. They cultured these fibroblasts in a collagen gel in that top chamber, that large open top chamber, and then the epithelial overlay on that. And they did this with 14 days of air-liquid interface and underflow. You can see here that we were able to get beautiful expression of the key markers for this model. What they were able to show is that they were able to certainly see a difference in the impact of these different fibroblast sources on the histological features of that epithelium. So again, the model recapitulated many of the histological features of metaplasia, including a range of differentiation fates that were observed from these different types of fibroblasts. And then CAF malignant regions significantly increase epithelial cell proliferation on chip and those morphological differences. So you can see that with this system, you're able to start to model these complex types of models that you couldn't do in vitro before. And in fact, if you go to the Emulate website, there's a wonderful webinar by Dr. Shimshoni on this model, and also how it compared to trying to do this on a transwell. And in fact, they were very unsuccessful in establishing the model on transwell. They needed that flow and that flow and that stretch as part of the microphysiological forces. So again, just to sum up, organ chips, we believe they can benefit every step of the pipeline, from early discovery, developing disease models that with closer to human gene expression and functionality to identify and validate drug targets. You're able, in lead optimization, to study these candidates and begin to rank order them by using complex systems that are able to really pressure test your lead candidates. You're able to move them into preclinical safety. You can predict diverse mechanisms of unexpected toxicity from preclinical drug candidates and assess human relevant toxicity observed in preclinical animal studies as well. And again, just echoing back to that predictive study that we did with drug-induced liver injury. And finally, in clinical trials, you can even look at, if you see a toxicity signal in clinic, can you go back and start to understand the mechanism of that on chip? And again, because we can recapitulate that complex biology, organ chips are a wonderful method for being able to do that. But this is all in the preclinical space. Organ chips, of course, have quite a large possibility of diversity of applications, including down the road, even taking patient material and really establishing patient on chip and looking at a patient-to-patient variation of response. And that's something that our organ chip community is actively looking to do right now. So with that, I would like to close up by saying that the organ chips themselves are available from Emulate. The way we deliver this is a full organ chip solution. We have the instrumentation, the Zoe culture module and Orb culture module. These are the instrumentation that you can buy and place in your lab. This keeps the organ chip biology and provides those microphysiological forces. We have BioKits, which have all that pre-qualified biology for those 6 organ molds that Emulate has brought to market. And also we have our basic research kit. These are blank chips where you can build your own model based on the biology and the cells that you're already using. So again, lots of ways that people can get started with organ chip technology. And in addition to these, Emulate also offers organ chip services, both for efficacy testing as well as toxicity testing on drugs. So with that, again, I would like to thank the organizers of the Avatar Medicine Forum for inviting me. And please visit emulatebio.com, and there's a rich repository of webinars, publications for you to really start to look at the science behind organ chip technology. Thank you.
Thank you, Luke, for sharing the connection and application of chip and data.